Every living cell faces a basic, terrifying problem. If it opens itself completely to the outside world, its contents scatter and it dissolves into chaos. But if it seals itself off entirely to stay protected, it starves to death. The solution to this paradox is a highly regulated border. The cell needs a barrier that acts like an impenetrable physical wall against external threats, while simultaneously operating as an open doorway for the specific nutrients it needs to survive. This diagram shows the foundation of that border, a structure known as the phospholipid bilayer. It consists of two sheets of molecules stacked back to back, with their circular phosphate heads facing outward and long fatty acid tails buried in the center. Those outward-facing phosphate heads are hydrophilic, meaning they readily attract water from both inside and outside the cell. Meanwhile, the internal fatty acid tails are entirely hydrophobic. They actively repel water and any charged particles trying to pass through. By combining a water-loving exterior with a water-hating core, the cell creates a highly selective filter. This chemical contradiction forces the surrounding universe to play entirely by the cell's rules of entry. Instead of a static brick wall, the membrane behaves like a flexible, undulating sea. The individual molecules are constantly moving, drifting past one another like objects floating on the surface of a crowded pool. Biologists call this the floated mosaic model. It gets that name because this drifting liquid is heavily studded with specialized proteins and lipid structures, scattered throughout the surface like tiles in a mosaic. Cholesterol molecules wedge themselves between the phospholipids to stabilize the fluid and prevent the membrane from tearing. Alongside them sit intrinsic and extrinsic proteins, complex structures that span either partially or entirely across the bilayer, acting as physical gates. This floating architecture sets the physical stage for cellular transport. It provides the necessary structure for the cell to either passively exploit the laws of natural physics or actively fight against them to hoard what it needs to stay alive. To actually bring resources across this border, the cell always prefers the path of least resistance. Whenever possible, it takes a free ride, moving molecules without burning a single drop of its own metabolic energy. This animation illustrates simple diffusion. Small, uncharged molecules naturally cascade down their concentration gradient, moving from areas of high density to areas of low density until both sides reach a perfect, even equilibrium. For polar or charged particles, that solid hydrophobic core would normally block them entirely. To get around this, the membrane provides a VIP lane called facilitated diffusion. Specialized channel and carrier proteins act as protective tunnels, letting these charged particles bypass the fat layer and continue their natural downward flow. In both of these passive transport scenarios, the cell survives effortlessly. It simply opens a door and lets the universe's natural drive toward equilibrium do all the heavy lifting. But that constant drive toward equilibrium creates an ever-present danger when it comes to the movement of water, a process specifically known as osmosis. Looking at this split-screen diagram, we can track exactly how osmosis works by measuring water potential. Pure water has the highest possible potential, set at a baseline of 0 kilopascals. When you dissolve sugars or salts into water, that potential drops into negative numbers, causing pure water molecules to naturally rush over to dilute the heavier concentration. If you drop a typical animal cell into pure water, the internal environment has a lower, negative water potential. Water aggressively rushes inside, building immense hydrostatic pressure until the delicate membrane completely ruptures, a fatal event known as hemolysis. Notice how the plant cell handles this exact same pressure. Plants evolved rigid outer cell walls made of cellulose. As water rushes in, the expanding internal membrane hits that strong outer wall and stops. Instead of popping, the cell becomes firmly turgid. For microscopic life, the physics of water potential dictate the difference between maintaining structural integrity and total cellular annihilation. Of course, cells can't always rely on the downward flow of physics. Often, a cell needs to pull in vital resources that are already highly concentrated inside the cell, meaning it has to forcibly push new molecules uphill against the gradient. To pull off this unnatural movement, the cell must pay a toll. It has to burn its own metabolic energy currency, a molecule called ATP. This animation illustrates direct active transport in action. An ATP molecule binds to a specific carrier protein in the membrane. The energy released by burning that ATP forces the protein to physically change shape, shoving the target molecule from an area of low density to an area of high density. Nature also engineered a highly efficient shortcut for this process, called cotransport. You can see this happening by the millions inside the human intestines, driven by a mechanism known as the sodium-potassium pump. Notice the two steps happening here. First, the primary pump burns ATP to violently eject sodium ions out of the cell, creating an artificial high concentration of sodium outside. When those sodium ions inevitably rush back inside down their steep mu gradient, they flow through a secondary channel, physically dragging a much larger glucose molecule in with them against the glucose gradient. Through active transport, the cell exerts control over its environment, engineering localized thermodynamic imbalances to hoard the exact resources it needs. Still, even these specialized protein pumps have size limits. How does a cell consume an entire bacterium, or export massive chains of protective enzymes that cannot possibly fit through a tiny membrane channel? To move objects that massive, the cell abandons the protein gates entirely. In a process called bulk transport, it weaponizes its own fluidity. Here we see endocytosis. The membrane physically folds inward to completely surround and swallow external material, pinching off to form a secure internal pocket. The reverse process, exocytosis, works identically in the opposite direction. Internal bubbles of material fuse with the outer membrane to dump their massive payloads out into the void. The fluid mosaic is so exceptionally adaptable that it can reshape its entire physical border on the fly to consume massive threats or distribute large-scale biological resources. When we pull back from all these individual microscopic pumps, channels, and flexing lipid walls, we get a clear view of the living cell as a holistic machine. The natural laws of the physical universe dictate that everything should spread out, dilute, and dissolve into entropy, but biology actively rebels against that fate. Life requires intense concentration, strict separation, and precise internal order. The cell membrane is the thermodynamic engine that makes that rebellion possible. By harnessing the free flow of physics when it is beneficial, and burning the intense energy of active transport when it must, the cell maintains the highly improbable balance that we call life.